So once again, welcome you all. I hope my voice is audible and the presentation is visible to you. Can you just confirm in the chat box?
Yes, ma'am.
Yeah, okay. So I can see some of you from the previous session and some new participants as well. So I welcome you all to the live session 2 on Environmental Biotechnology course. And I am Guru Priya, a research scholar in the Environmental Engineering Division, Department of Civil Engineering, IIT Madras. So, as discussed in the first session, we would discuss the course contents in a question and answer pattern where all of us will first discuss the answers in the chat box. All of you can answer. And finally, I'll also share the answers that I have prepared. So, in today's session, we'll have both short answers as well as some multiple choice questions as well. So we'll get started and let people join us. So the first question for today would be to define species. So in your own context or own terms, you can just define species in the chat box and then we'll have a discussion over it. Okay, so we have a handful of answers now. Species is the basic unit of biological classification. They are reproductively isolated organisms. Yeah, and they can mate with themselves. A species is a group of microorganisms with similar genetic and functional traits, and they are used for environmental application like waste treatment or bioremediation. Okay. large groups of organisms capable of interbreeding and producing fertile offspring. So we have different definitions in terms of the context. Uh, Most of you have given all of the answers, and many of them are similar also. So, ideally, species is the fundamental unit of biological classification, and it helps us to describe, understand, and compare the biological diversities. So, when we are grouping into species, we are seeing out some similarities in those organisms and grouping it as species. So, when we have different species, we are able to look at the biological diversity in that. And most importantly, in our context, when we talk about this environmental biotechnology course, microorganisms are going to be the main topic of this course. So, in that case, defining a microbial species, microbial species is defined as two strains with 70% similar hybridization in their purified genomic DNA. So, if there is going to be 70% of hybridization between the purified genomic DNA between two strains, it is going to be equivalent to 94% of nucleotide identity in the whole genome scale. So, by which we say when they are genetically similar, the concept of genetic similarity, as one of you had told, comes into picture here. So, this genetic similarity defines a microbial species. So when the gene is going to be similar, the functions or the activity also is seemingly correlated and said to be similar. So coming to this microbial species definition, there is this definition with some numerical terms of 70% hybridization in their purified genomic DNA.
I guess I love you. What wouldn't that?
Mute yourself. Okay, so when we are going to talk about this 70% hybridization or 94% nucleotide similarity, we are going to analyze the genomic DNA of the microbial species. So how are we going to determine the 70% hybridization? And when we are talking about a definition for something, we need to know each and every term in it. So is there any idea how are we going to determine the 70% hybridization? And any answers related to the method, um, or your understanding of what this could mean is welcome. Not just the exact procedure I'm asking, I'm just asking an overview of what does the 70% hybridization mean and how can we determine that. So that will be your next question.
Hat selection, ma'am.
Can you repeat yourself?
Hat selection.
Okay. Mahalakshmi, may I know what you want to clarify? Do you want me to repeat the question?
Yes, ma'am.
Okay, so in the lecture you would have heard that microbial species, when they're defining microbial species, there is this 70% hybridization that has been talked about. Like when two strain, when we are comparing the genomic DNA of two strains, and if it is 70% hybridized, uh, that equalizes to 94% nucleotidic identity between those two organisms, and hence we tend to classify, okay, these both organisms are going to fall under the same species. So that was what the definition that was there, right? So based on that, how are we going to determine the 70% hybridization, or what that means? Ideally we could perceive something as some genetic similarity we are looking for, right? Okay, Akshaya Shri has answered that we can determine them by different types of hybridization such as DNA, and DNA, RNA. So here we go with the answer. Right, first we have to denature the DNA. So first step would be to, if people have read about techniques like PCR, polymerase chain reaction, it is something similar, I would say, not exactly the same, but it's something similar. So if for the people who are unaware of these techniques or how to determine this nucleotide sequence and all, the step is that you isolate the DNA and it undergoes fragmentation and denaturation. So the genomic DNA from both the strains, that is the both the organisms that we are considering, we are going to extract the DNA and it is fragmented into small pieces. Then we are going to heat this DNA. We all know that DNA is double stranded, so when we are going to apply heat, it is going to separate into two single strands. So the DNA from one strain is labeled, then mixed with the unlabeled DNA from other strain. So the strain, the labeling is going to be done either through radioactive methods or fluorescent dyes. And then we are going to allow these strands to re-anneal. So this step is called this hybridization step. So when the mixture is cooled, the complementary DNAs only can realign to form a double-stranded structure from all the single strands that are present. So we will then check upon the extent of hybridization. So only when both these DNAs are going to be similar, uh, the strands are going to interchange and get hybridized. We are not directing the hybridization anywhere, right? So one strand from the strain A and one strand from the strain B can hybridize. We will be measuring this extent of hybridization. So for this measurement only, we are initially labeling the DNA from one of these strains using radioactive or fluorescent methods. So based on detecting these radioactive or fluorescent compounds, we will know the extent of hybridization. And then finally we are interpreting that if we have 70% or more of the DNA under hybridized conditions, the genomes are similar, and we can call them as organisms falling under the same species. So when I'm going to give you two strains A and B, unknown strains, and asking you whether these belong to the same species, this is what you will be carrying out. Is this clear? Is this clear or should I repeat myself? If this is clear, just let me know in the chat box so that I can move to the next question.
Clear, ma'am.
So the next question is to compare species diversity and species richness. Okay, so we have different answers. Diversity means the variety and variability of species in an area. And species richness refers to the total count of all species in a specific location. Okay, whereas species diversity is the count of each species in that area. Then why are we mentioning it as species diversity? So we are just talking about how uh, diverse the species found in a particular area are. So in that context, I don't think species diversity would be the count of each species in that area. We are talking about a diversity of the species, which means we are considering more than one species. So the next answer is species diversity refers to both number and abundance of species, while richness only counts the number of different species. Richness, number of different species, and diversity includes the concept of abundance, right? So the last two answers are perfectly fine. So ideally, total number of different types of species present in a particular area. This is what both species diversity and richness talks about. So in both the cases, we have different types of species present in an area and we are describing some aspect of it. So as rightly mentioned, while defining the species diversity, we'll mostly have both the number and abundance into picture. While talking about the richness, we'll count only the number of different species present in uh, their system. So we can term that species richness is a component of species diversity, because in diversity we are talking both about the number and abundance, whereas in case of species richness we are going to talk about only the number in most cases. But also in some places you can see these two terms are used interchangeably. So they are very close to each other. There is a minute difference where diversity also talks about the abundance. That is why the question was to compare diversity and richness. So, on summary, you can remember that species richness is a component of species diversity. So, with that, the next question would be to differentiate microbial population and community. So, in the last question, we compared species richness and diversity, and now we are going to differentiate microbial population and community. And also, if the discussion for any of these questions is not satisfactory to you, you can again post it in the chat box so that I discuss over it once more. Okay, so population is includes all members of a particular species present in an area. Same, it includes a group of the same species. Whereas in the case of community, it consists of multiple interacting species or more than one population. So exactly, population is a group of microorganisms of same species residing in the same place at the same time. And in case of community, it is a group of population, right? The catch in population is mostly we define population as group of microbes of same species generally, but the clause also mentions that they have to reside in the same place at the same time. So, and that is when we are going to call them as a population. So the next time you define them, you also try to include these terms. And the next question is regarding the significance of species abundance. List down the significance of species abundance. And before listing down the significance, if someone wants to define species abundance, that would also be great. You can define species abundance and list down the significance of it. Why is it important to study species abundance, or what inference species abundance, the study of species abundance gives us? All these is what I'm asking in the name of significance of species abundance.
Ma'am, species abundance refers to the number of individuals of a specific species which is found within a specific area.
Okay, right. Species abundance shows how many individuals of a species are present. So with that data, we'll be able to understand the ecosystem health, balance, and biodiversity. And the other answer was richness does not clearly depict at what degree the area is crowded. So in that case, we need the concept of abundance to know how many number of microorganisms in each species are present. It refers the measures of individual and common area of ecosystems. Okay.
And it is also important for understanding the structure and function of ecosystem, ma'am.
Okay, right. So now we know that species abundance refers to number of individual organisms in each species present. So that is what we mean in different terms as how crowded or how many number of microorganisms are present in that. And we have also seen that it helps us understand how the ecosystem functions. So as rightly mentioned, by knowing the species abundance, we will be able to calculate the proportion of each species. So when we know the proportion of each species, we can understand the organization and functioning of the ecological communities. As in, when we have two different species of organisms which do different metabolic activities, we'll try to find out why both of them coexist or what is the effect of both of them coexisting in that ecosystem. And finally, the one point that all of you have missed out is that the species abundance can also be an indicator of the interspecies interactions. So we know that if there is a competition or predation in that ecosystem, like when we are going to study the ecosystem over a period of time, we can find this out. So when we are going to have a very new ecosystem formed or we are following the ecosystem over a period of time, then we would get the information on interactions like competition and predation. If on a given day I'm going to study the species abundance in any ecosystem, then it's just knowing which species have a upper hand, and even with that you can little bit try to infer on what is the relationship of that particular species with other species and why it is going to outcompete the growth of other species. And if I'm going to regularly monitor it over time, I can confirm that there is some competition or predation in that ecosystem. Next is a very simple question. Which genetic marker is predominantly used to define microbial species composition? Okay, enough of answers supporting 16 is rRNA, and that is the genetic marker that is predominantly used. The next part of the question, someone has already started answering that it is present in all bacteria. What are the other reasons why we use 16S rRNA as the predominant genetic marker in microbial species? So one reason, as stated, is because it is present in all bacteria. And is it only bacteria, or there are also other organisms in which we can use 16S rRNA as genetic markers? Also in archaea. Okay, right. So for those who uh, are from a very different background, uh, apart from microbiology, there are microorganisms. Generally, does not mean only bacteria. There are also other organisms like archaea. So the basic difference between bacteria and archaea is that archaea can grow in extreme environments. So that is how they were discovered, and they were structurally found to be different from bacteria. Like their cell wall composition and everything was different from bacteria. So archaea and bacteria are not the same. They are two different types of microorganisms present in nature, and both of them have 16S rRNA in them, and that is a genetic marker. Any other reason? Okay, it has polymorphic regions, and what is the benefit of having polymorphic regions?
To distinguish between different bacterial species, ma'am.
Okay, we are using this to differentiate between bacterial species, that is right. Polymorphic regions are different in different bacteria, and we can easily distinguish them. Okay, one more answer is that using 16S rRNA is most accurate and reproducible. Right. So to sum up, we predominantly use 16S rRNA as a genetic marker because of its universal presence. So all bacteria and archaea possess the 16S rRNA. rRNA is the ribosome RNA. Ribosomes are the protein factories of the cell. They are generally termed as the protein factories of the cell, and they have this RNAs. Since it is present in ribosome, we call it as rRNA. And in the small ribosomal subunit, we have a gene called 16S rRNA gene. it is universally present. So it is not like we are selectively using it for some organisms and not for the others. It's universally present in almost all prokaryotes, and especially bacteria and archaea, which will be of our major interest. And they also have conserved regions and variable regions. So conserved regions, in the sense, these conserved regions are going to be the same. You imagine rRNA to be some sequence like ATGC or AUGC. It is going to be some sequence like that. So in that case, some regions of the sequence is going to be conserved or same in all the different organisms, and some regions are going to be variable. And this variability helps us in carrying out this taxonomic resolution. So using these conserved regions, we can multiply the genes. If you're going to design a primer for all these conserved regions, it's going to bind to the conserved regions, and multiplying the DNAs into multiple copies is going to become easy for us. And then using the differences in this variable region, we can find out the genetic similarity and differences by sequencing it. So that is why we use 16s rRNA as the genetic markers in identifying prokaryotes, especially bacteria and archaea. And other factors include that the evolutionary rate is very slow for 16s rRNA, and hence it is more reliable. And also, the length of 16s rRNA is approximately 1500 base pairs. So, it is kind of an optimal length for sequencing studies and informatics studies. We will neither have too much information or too less information. So, these are the reasons why we go for 16s rRNA sequencing to identify the microorganisms. Is that clear? Okay, so in case you have any doubts with this question, please post in the chat box. I'll repeat again. In the meantime, we'll also look at the next question, which is also a simple question. In an oligotropic and extreme environment like acid mine drainage, you will find what kind of organisms?
Option B, ma'am. Low species richness and high abundance of few species.
Okay. I'll wait for one more minute for other people to think and respond, and then we'll. Okay, so B is the right answer. Oligotropic and extreme environments. So that means not all microorganisms are will be able to grow in that. It's going to be extreme, and only the organisms which are going to resist that extreme conditions can grow in that area. So in that case, the species richness is going to be very less. Richness, or in terms the diversity, not many kinds of microorganisms are going to be present in that region, so it is going to be low species richness. And then high abundance of few species, we know that only few species are going to be present. Abundance, we are studying about the relative proportion of organisms in that, right? So in that case the abundance is going to be high. It is going to be interchangeably same only. Because we have low species richness, there is going to be high abundance of few species. We can also interpret it that way. Right, not many species can grow there. Only the organisms which have the resistance to grow in some extreme environments can grow, like acidophiles, halophiles, barophiles, thermophiles, as we can list down. But yes, the nutrients are available there. Resources may be available, but the physicochemical conditions will not support high diversity of microorganisms growing there. And the next question is regarding what is a niche of a microorganism referring
Option B, ma'am, all interactions and roles within its community.
to. Okay. So, as mentioned, the niche refers to all interactions and roles within its communities. It's not pertaining to any particular nutrient source or physicochemical conditions or size, but it generally refers about the interactions and roles in a community. So the next question is, which of the following best describes high species richness but low abundance? Again, kind of a repetitive question, but reading the options can be.
Again, option B, ma'am.
Yes. So because it is going to be about high species richness, the term says that it's going to be either B or C. Many species are going to be present, not few or only one species, but we are talking about low abundance. And C clearly states high abundance. So in that case, many species, each with low population, describes high species richness with low abundance. So yes, so these are the reasons. We have already discussed all of them. The reasons include universal presence, the variable and conserved regions, and there are also other factors like some developed technologies are there for studying these and all this. So next, moving on about the guild formation. What is the fundamental basis for a guild formation in microbial ecosystems?
Option B, ma'am.
Yeah. So is the fundamental basis for guild formation is metabolic linkage and relatedness. So metabolic linkage as in states that The resource utilization and the product protection from each microorganism is the metabolic linkage between the organisms. It is not about taxonomic similarity or a geographic proximity that aids in the formation of a guild. How functionally similar microorganisms are, that leads the formation of a guild. Okay, so now define guild. So we know that functional similarity is defined is the deciding factor for guild formation. So based on that, define guild.
Any group of species exploiting the same resource, ma'am.
Okay. So group of species that uses the same resources performs the same function. So yes, a group of species that utilize the same resources in a similar manner. So we talk about both resource utilization and the function, and here it is regardless of their taxonomic relationship. So what information does a guild decipher in our study is that it tells us about the functional redundancy. It tells about how multiple species perform similar functions. So this gives the ecosystem the stability, not all going in all different directions, but different microorganisms, like they either metabolize the nutrients present in the ecosystem and they produce similar products. So this gives a stability to the ecosystem. And next, the metabolic cooperation. So there can be relationship among different population or different species in a guild that makes each other survive. So sometimes there can be competition. Sometimes it is that the one organism utilizes the resources and produces some compounds after fermentation, and that again is useful for another set of organisms to survive there. So that kind of metabolic cooperation is possible, and we can study using the information on guild. And next, we'll be able to predict responses, like a guild level response to environmental changes can be predicted when we long time track how the guild behaves, or how the individual population in the guild behaves. We can predict the responses to environmental changes, either natural or anthropogenic, can be predicted. And the next thing it deciphers is the management targets. So we can identify the key functional groups for ecosystem manipulation or restoration. So when there is something wrong with the ecosystem and it is deteriorating, we can find out what is the key aspect that we have to focus on in order to restore the ecosystem. We need not focus on very holistic development, because our urgent need would be to restore the ecosystem. And we can also manipulate the ecosystem. If something is not looking very sustainable about the ecosystem, we can try to manipulate it on targeting some functional aspects, key functional aspects in the guild. And this we would be knowing only when we know what is the common resource that all these microorganisms are utilizing, or what is the common product that all of them are producing. And the last inference that we would be able to collect from the information on guild would be the knowledge on system resilience, recognizing how guild interactions maintain ecosystem stability. So initially we would know that there is going to be some ecosystem stability, and we could recognize how this function happens. And this can also help us develop processes in lab, like in reactors in lab, for environmental remediation elsewhere of sites, or also in terms of waste treatment or similar processes. So this is the significance of knowing about guilds. And the next question is to compare and contrast the three main energy acquisition strategies in microbial ecosystem. You can perceive the question as you are able to, and then give the answers. And I'm repeating myself that if you have any other doubts, you can always post it in the chat box, and we can discuss that in between also. Okay, so as rightly mentioned by Akash and Mahalakshmi, so we have phototrophy, chemotrophy, and organotrophy. So, energy acquisition strategies, in the sense, any metabolic activity in our system, we need energy. So, primarily for all the breakdown reactions in our body, we need energy, and this can be derived from different sources. So, like in case of photosynthetic organisms, which synthesize their own food, they use solar energy, light energy. Photo means light, and this is predominantly going to be solar energy, and they are going to fix the atmospheric carbon dioxide into organic matter, like carbon dioxide to glucose, which is an organic matter. So this kind of conversion is phototrophic conversion. So the energy is derived from light sources. And in case of chemotrophy, we have chemoorganotrophy and chemolithotrophy. So here the source of energy is going to be a chemical compound, and that is why we call it as chemotrophy. In case of chemolithotropy, litho means from inorganic substances. So in that case, inorganic substances like hydrogen ion, hydrogen sulfide, ammonia, all these can act as the energy producing components. They use the chemical energy from oxidation of these inorganic compounds, and then they fix the carbon dioxide. So in this case, since they are going to synthesize their own food or organic matter, and only the source of energy is going to be a inorganic compound, we call it as chemolithoautotrophic organisms. And in the case of organotrophy, the energy is going to be derived from organic substances using organic carbon. So in all the other cases, the energy source is something, and the carbon source is going to be carbon dioxide. Here both the energy and the carbon source is going to be the organic molecule, carbon containing molecule. So that is why this is called organotrophy, and it includes heterotrophic organisms that decompose organic matter. So in terms of ecology or study of ecosystems, all three strategies are going to contribute to the ecosystem function, where the autotrophic processes mainly are going to be the production processes, and the organisms that carry out this are going to be called producers, while in the organotrophy, the consumers and the decomposers are going to be present. I hope many of you were not able to answer this because you are not able to identify what may energy acquisition strategies mean. So the energy source is and the carbon source are key for all metabolic processes in living systems, especially also in microbial systems as we are studying now. Another simple question: which of the following represents allochthonous organic matter? Okay, we have D, we have C. Okay, so most of you go by option C, and C is the right answer. The term allochthonous by itself means uh, a, an object or a formation uh, that has originated in some other place and it is currently found in some other place. Let's say you're finding some object in a particular place, but the origin of that object is not the place that you found, but it is somewhere far away. This is the meaning of the term allochthonous. So, allochthonous organic matter is going to be something externally derived organic matter from agricultural runoff. So, uh, the point of origination is going to be something different, and because of a runoff, it has been carried away from the point of origin to the current position where we have found out. So that is why externally derived organic matter from agricultural runoff represents allochthonous organic matter. Whereas in the case of organic matter synthesized by phototrophic bacteria in situ, the term in situ states that it's originating from the same location, and metabolites released by local microorganisms. So when local microorganisms are going to synthesize something, it is not something from a very distant origin. And dead microbial cells are also from the same ecosystem. Like the organisms that are dead there are going to be present there, and that is not allochthonous. So, and now easily the term autochthonous organic matter refers to very easy question. I've already told the answer, so if I get few answers, I'll just move on to the next question. I would reiterate the question. The term autochthonous organic matter refers to. Anyone else left to answer, or can I move on with answer? Okay, so in case of allochthonous, we defined it as an organic matter present in a particular location that we have found out, but the origin is not the same location. But in case of autochthonous, it has to be organic matter synthesized in situ by local organisms. It is not externally derived. If it is going to be externally derived, it is going to be autochthonous. And it is not only dead organic material, it can also include dead organic material, because microorganisms are going to be present to that particular region, and once they are dead, that organic material is can be called as autochthonous. And we are talking about organic matter, and it is not inorganic. So in that case, organic matter synthesized in situ is going to be the answer. And any clarifications you want regarding the answer? I see many of you answering A. The term autochthonous refers to indigenous, something that is being generated in the same system and found in the same system. It is not being carried away to another place or habitat. So, in that case, autochthonous organic matter is synthesized in situ by local organisms. In situ means inside the system. So, in that case, that is the answer. I hope you are convinced. If not, please let me know in the chat box. I can come back and repeat myself. For now, I'm moving to the next question. Bhavna, were you asking me to clarify something, or uh, are things fine? Mm, rightly mentioned. Allochthonous means external. Autochthonous means inside. That is indigenously present. So that is why the answer for previous question was, when we're talking about allochthonous, the answer was externally derived organic matter from agricultural runoff. Whereas when we were talking about autochthonous, it is organic matter that is synthesized in situ by the local organisms, or it is synthesized in the system by the local organic matter, local microorganisms. Sorry. And now moving on to this question. Is this question so tough, or are you all held up with the previous question only? Okay, so this is kind of a memory based question, and the answer for it would be the guild 4 predominant is predominantly composed of methanogenic archaea. So interestingly, in all these options, this is what I was trying to clarify when we are talking about the 16s rRNA gene. There are two different types of microorganisms. One is the bacteria, next is the archaea. And archaea, mostly they will be methanogenic. Not all archaea are methanogenic, or not all methanogens. Only methanogens consist of archaea. It's not that way, but mostly it is methanogenic archaea that we tend to uh, look upon when we are talking in terms of environmental biotechnology. So we'll move on to the next question. The primary producers in microbial ecosystems are characterized by which of these? And again, I feel this is one other easy question. So as rightly mentioned, synthesis of new organic carbon from carbon dioxide uh, characterizes primary producers. So as we were talking about the energies acquisition strategies, we were talking about uh, phototropes, chemotropes, I mean chemolithotropes, chemoorganotropes and all. So I was telling about there are two things. One is the carbon source and other is the energy source. So in terms of energy source, it could have been light source or organic matter or inorganic matter. But in case of the primary producers, the carbon source is going to be either the only the carbon matter, right? It's going to be carbon dioxide or any other compounds like glucose or starch. uh, so in that case, synthesis of new organic carbon from carbon dioxide is going to be the characteristics of primary producers. And again, to answer this question that was previously answered, and I hope you are a little bit stuck with this, so I'll just move on right away with the answer. So in the case of anoxic zone, the guild 1 is going to be fermentative bacteria, and the guild 2 is going to be denitrifying bacteria. Guild 3 consists of the sulfate reducing bacteria, and guild 4 consists of methanogens. So first we have the fermentative bacteria, where it is going to consume the carbon matter present there, and then do fermentation. And the denitrifying bacteria is going to work on the nitrogen cycling there, and this also doesn't require much of oxygen, so that is why it is present in the anoxic zone. Similarly, sulfate reduction also happens in the anoxic zone. And finally, methanogenesis, which is um, the production of methane. It is mostly strictly anaerobic. So anaerobic is the condition without air, and anoxic is without oxygen, so more or less they mean the same term. So in that case, we have all these different groups of organisms present in different guilds in anoxic zones. And the next question is, the guild formation facilitates ecosystem function by which of the following? So we all understand what is a guild, right? A guild is a group of species that utilize the same resources in a similar manner. So thereby they form a guild. So how this guild formation facilitates the ecosystem function? Uh, when we see about the first option, does guild formation reduce competition between species? Maybe, but there are very less chances. So in it is not going to eliminate the need for abiotic factors. Abiotic factors include physicochemical conditions that require for the growth, and it is not going to simplify energy transfer much, right? So it is explicit that a guild formation is going to coordinate the metabolic processes through population interaction. Like a population that utilizes the same resource in a similar manner is going to have a communication within itself, and similar kind of guilds, different guilds present, are going to work in an orchestrated manner. So keeping that in mind, enabling coordinated metabolic processes through population interactions is the key facilitation from guild formation in any ecosystem. And so this is the second but last question for today's session. We'll just have one more question for answering and then we'll wind up. Chemolithoautotropic bacteria contribute to ecosystems by which of the following? Okay, so as discussed, it is not going to use decomposed matter or ferment. It is not only going to perform fermentation, or it is not going to be only in aerobic conditions. As we saw in the previous slide about that gills in anoxic environment, all this nitrification or sulfate reduction are all kind of chemolithotropic processes. So in that case, it is not only happening in aerobic conditions. We know that they are going to reduce inorganic materials and as electron donors and then produce organic matter, and hence we call them autotropic also. So the first part, reducing the inorganic material, gives the part of the name of chemolitho, and producing new organic matter, they become autotropic bacteria. And the last question for today is, in microbial ecosystems, the flow of energy and nutrients is achieved through which of the following? Okay. So as rightly mentioned, flow of energy and nutrients is through coordinated function of populations. It's orchestrated. It's not very random, or it's not about competition. And abiotic factor, only abiotic factors. Abiotic factors also contribute, but it's not wholly about abiotic factors. So with that we can close today's session. And if you have any doubts, you can post in the chat box. And also let me know if this pattern of session is okay with you. Yeah, you can also unmute yourself and ask if you have any doubts. So please let me know if you have any doubts. If there are no doubts, please give a thumbs up so that I know that you don't have any doubts. Okay, thank you for the response. And the next week also we'll be continuing with the same pattern. Maybe I'll try to include 10 minutes, 10 to 15 minutes of presentation either before or after the session regarding summarizing the whole week's content. So with that, thank you all for joining today's session. If there are no further doubts, you can feel free to leave the session. Okay, so Bhavna, your question is allocturnus means external. Uh, what do we mean by external and what are included in it? External means like we have one habitat. Whatever nutrients are already present in the habitat, those we call as internal or autochthonous. Whatever comes from outside, that we call as allochthonous. For example, when we were talking about that agricultural runoff, the nutrient was already present elsewhere, and because of the runoff, the nutrients has been carried out to this particular habitat. So in that case, we call it as allochthonous. It has been carried away from a different place to this place. In case of autochthonous, it is going to be already present in that place only. Like whenever first time microorganisms are colonizing some place, some indigenously something is present in that place, and after the initial colonization, what happens is this microorganism is going to consume the resource, and then it is going to produce some other organic compound in that place. So that product that it produces is going to become the autochthonous organic matter uh, after the initial colonization. And finally, what happens at one stage, this microorganism is going to die. So the dead microorganisms, like when we are going to have a dead animal or plant, when we're going to bury it, it is those cells can be taken up by other microorganisms. Similar way, when another microorganism die also, that can be taken up by other microorganisms, and the microorganism is indigenously present in the system, and that is why we call it as a autochthonous organic matter when we have a dead organic matter. So this is a difference between autochthonous and allochthonous. Anything that has been produced in the system or been initially been in the system, we call it autochthonous. All others which has come from some other habitat, we call it as allochthonous. Is that clear? Bhavna, is it still confusing or is it clear? Okay. Okay, so thank you all for joining. If there is some any other doubts, please let me know, or else kindly let me know if I can close the session. Okay, so if there are any other doubts, thank you for joining. We'll meet in the session on next Tuesday from 7 to 9. Thank you all for joining. I'm now closing the session.